Hello, and welcome to this third season of This is Epigenetics, the bilingual scientific podcast presented by the Canadian Epigenetics, Environment and Health Research Consortium, or CEE-HRC podcast, hosted by their trainees. We interview renowned Canadian scientists to talk about their career, their research, and of course their passion for epigenetics. We hope you enjoy this episode. Hi, my name is Sabrina. I'm a PhD candidate in the Couture Lab at the University of Ottawa, and I will be your co-host for today's podcast.
And I'm Dr. Laura Godfrey. I'm a postdoc in Dr. Scott Armstrong's lab at the Dana-Farber Cancer Institute in Boston. And today it's my pleasure to welcome Dr. Amélie Fradet-Turcotte from the University of Laval, who's going to be with us today to talk about her career and research in her lab. So welcome, Amélie.
Hi, Sabrina. Hi, Laura. Thank you very much for having me. I'm happy to be here.
So yeah, maybe we'll start with a first question, which is, we're just interested generally, what made you interested in science, and why did you decide to become a researcher first of all?
Yeah, so I was interested in science, I would say, when I start my my bachelor degree. This is where I realized that I cannot deal with learning everything by heart, but I really love learning how mechanisms work and trying to understand this. And this is still what I'm interested to in different type of science we're doing in the lab. So when did I decided to become a researcher? It came naturally. I did not decide to become a researcher at the beginning of my study. I liked doing research. I went to different internship and different lab and see what I don't like in terms of like working. I don't like to work with animals, for example. So I learned what I don't like to do and what I like to do. And at some point, it came out that I had an opportunity to interview to become a PI. And I said, oh, well, I like to have an intern, to show out, to transfer my knowledge. So let's try it. And this is basically what I've been doing, trying it. And it's working fine so far.
You mentioned you're going into being a PI, but what was your professional journey, before that? How did you go up the ranks to get to that point?
So I went to University of Sherbrooke at the bachelor degree, and this university have a co-op program. So I really start early in my career and doing internship. And I was working part-time at the beginning, so my grades were not like excellent. So my first internship was in an industry that was studying the composition of the metal. So that was really more chemistry part of it. And then my second internship was in a lab that studied the toxicity of bacteria to potato. So that was really different. But then I was a really short internship in University of Sherbrooke. And that opened the door for the third internship at Boehringer Ingelheim, which is a pharma that was before in Montreal, specialized in studying viruses, oncoviruses, such as hepatitis C, human papillomavirus, and we also work on HIV. So I did 2 internships there. So that really gives me my first contact with the study of viruses, and I became fascinated by the fact that viruses such as HPV, have 8 protein. And they take over your cell and they lead to the development of cervical cancer or pharyngeal cancer. So I was kind of fascinated to understand how so small amount of protein take over your cell. And then there I met my, actually the person who became my mentor as a master degree and PhD degree, Jacques Archambault, and then he opened his lab. So he quit industry in the late age and started his own laboratory in Montreal at the RRCM. So that was quite of a special transition that you don't see that often, I found. So he opened his lab there. I opened the lab with him and spent about 6 years there. And then naturally my project, so I was working there on a human papillomavirus replication. And we started to see that there was an association with the induction of DNA damage response in the cells that were infected or expressing the viral protein. So I got interested in these different pathway, and this is where I decided to pursue my postdoctoral fellow in understanding these pathways. So everything dealing with genomic stability, but from the cell point of view. So I used to do a PhD on about 8 protein and then switch back to the cells. So that was quite a big move at the time for me. But I really loved my postdoctoral time there in Daniel Droché's lab at the Lunenfeld Institute in the Research Institute in Toronto. That was an amazing time with tons of really talented postdoc and PhD students, all really keen into pushing science to another level. So that was a really good time. And then this is where, after a couple of years there, where we had our first big story, and then I met a director from University of Ottawa who was looking for PIs and then offered me to apply. And this is where the journey started to do application, interview, and that took about a year. So I moved after 4 years total in postdoc, I moved to my position here in Quebec City. So I started with Ottawa, but interview a bit everywhere, and decided to come here because of the core of people that are working on chromatin and DNA damage, which are two main domains we also pursue in the lab, trying to understand how that works.
So you said with your internship, you did have experience in industry. What kept you going in academia? What did you like, dislike from both academia or industry?
I would say that industry is it's an imposed subject, while academia is the freedom. Both of the industry and the academia are high stress, high demanding job, but on different level. And what I experienced in Boehringer, I loved it. But as a PhD student or master degree, my job would have been to do an assay for 10 years, set up the assay, and then screen for 1,000 of different compounds in the same assay. And I didn't find that really exciting. And I could see how my director was stressed at the moment, because in academia, you do have deadlines, but in industry, you also have deadlines sometimes to present tomorrow a result that you don't even have. So this was a stress that was not fit for me. And the other thing that I was not really excited about is that as a biochemist, I was doing the assay, but in industry, a huge part of the design or the excitement comes with new molecules. So there's a lot on chemist, and then you're kind of just testing the molecules. So that was the feeling at the time I had that prevented me to stick into biochem. And in academia, well, it's a total freedom, right? So it's come with the stress of writing grants. But I decided to embark this journey with the feeling of, I'm going to try, I'll do my best. And if it doesn't work, I can do something else. And I always have this kind of mindset. I do it because I like it. It's fun for many reasons. But if it's become unbearable, then I try to do something else.
Could you explain a bit more what was your research during your postdoc? Was it similar to your PhD work, or was it a completely different path?
Completely different. So during my PhD, I was really studying the viral protein E1, which is a helicase that assemble onto DNA, open the DNA, and then take all the factor from the cells to promote replication of the virus. I was really looking at one specific enzymatic event. However, in a postdoc, I work on genomic instability and really went to try to understand how protein, so in that case was 52BP1, I was studying, but a factor that could recognize modification of the chromatin that occur on the chromatin right after a DNA damage. So after a DNA double strand break, there's a series of events that occur in the chromatin. It's going to be phosphorylated, it's going to be ubiquitinated, and there's protein that actually read this signal and are specifically recruited. And the order at which they are recruited would actually drive how the cells will decide how to repair a specific type of damage. So that was really the whole goal of my project, try to understand. We knew that this factor, 52BP1, was going to the chromatin, and in a manner that was dependent on the addition of a ubiquitin. But we did not understand how the protein was reading it. So we really used biochemistry approach and cell biology approach to tackle this question. And that was the big discovery in my postdoc, understanding how two domains of a protein bind to modification at the same time, and this specificity allowed the specific recruitment after damage. So more biochemistry based study that we after validated in the pathway or DNA repair pathway in cells using microscopy and different types of approach.
And maybe, going back to your journey so far as a PI, what do you enjoy, about being a PI now? And how was the transition from a postdoc to setting up your own group? And what are some of the challenges and some of the things you've really enjoyed so far about it?
Yeah. So part of the reason I chose to come in Quebec City was actually the community that was really bound together. So that I mentioned it because when you arrive somewhere, this community and the mentorship is important, because you go from being a postdoc at your bench to ordering machine for your lab, recruiting people that you've never done before. So you need to have people helping you into this process. So what I like about the job is the diversity of the job. You do your science. This is one portion of it, but you also discuss science, different type of science with different people by being on committees for students, and you teaching. So you actually do class. You reviewing grants on panels, so you see how people write. You discuss different type of science. You see what it's about. So it's really opening your mind compared to postdoc, where you're like really narrow on what you want to do, what you want to publish. So the diversity there is amazing, and the students are really amazing. So you recruit students who start, and you always have to remember that you know nothing at the beginning, and then you start and you climb up this hill, and being so proud of seeing them moving further into. I basically have PhD student in the lab, and then I see them moving to postdoc and following their success, and I'm really happy about that. So this is part of the job I really enjoy. The challenge is you don't know what to do in terms of ordering or at the beginning, or how you should lead the lab. But I mean, you find as a postdoc, you learn how to find solutions. So this is not different. You find solution, you find people to talk to. And the biggest challenge is to say no. And I guess it's on a different type of job, but especially here, you have different people asking you from the university side to teach or to be on different committee. Or I'm also working in a hospital, so there's other demands there, and there's people in the lab who have demands. And so how to organize and say no and prioritize is really important. I also have 2 kids, so it's kind of really important to know where is the limit. So this is a work in progress, but I try to stick to the mentorship I have. And we also created a couple of years ago a website. It's not a website, it's a Slack for women in science. And we focus more in Quebec, a bit Ontario right now, but we're about 135 women who discuss challenges of how to train students, how to navigate the academia, and what are the different challenge. And I think it's important because most of my mentor have been male, and it's really good. But we also sometimes think differently, and having this different sets of opinion really did a difference in my life and how I make choices.
That sounds really nice. There's also, you have mentors that are like more senior to you, but like having people at the same kind of stage too, who are women, experience the same thing sounds really, really cool.
It's really important, actually. So you actually feel the normality. Having kids, been surrounded by people who either have and don't have kids at the moment, who are older. It really helps you to navigate how to deal with that, how to say no, what you can have access to, and whether your way of actually mentoring is maybe different, but it's okay. It's just another way of mentoring. So I think we do need these different models. It's really important because science is a lot of fun, and it's really suitable with family. So I think this needs to be advocate more, because I didn't know it as a postdoc, and I think the chance that it would work, and it did. And talking to women, it do work.
Maybe just staying into that work-life balance. How do you balance work and your personal life with 2 kids?
I mean, academia is amazing for that. So that's one of the things. Being in academia is like having your own industry. I mean, your own business, but with a safety net if you have a tenure track, right? How do I balance? Well, priority is family for sure. If something's wrong, I can leave whenever. But the good thing is that I can leave at 5, going and play with the boys, and then they go to bed, and I can go back and finish my whatever I'm doing. And it seems like I'm working all the time, but this is my own thing, right? So it doesn't feel like when I open my computer at night, it doesn't feel like, oh, I need to do my hour, it to be done. I mean, some stuff we don't like to do, completing forms, stuff like that, but looking for your research, talking to students on a channel like Slack, for example, helping them answering the question if they're stuck. It's really, I can do that whenever. So it's really flexible. So I would say it's easy. Do I have hobby? I mean, that was one of the questions. No, I don't have hobby. Hobbies are my kids and my friends. I go and see my friend. This is the way I unleash. I have a lot of friends who are not scientific, so they don't care about chromatin and experiments. So that's really also, I would say that these are my hobby, but I'm not squeezing in something more in addition of that.
Maybe just going back to your professional journey, did you have any role models or scientific heroes as you moved through your career?
I would say I had many. I admire all my mentors for their for many reasons. My PhD supervisor was amazing as taking time to show me how to write, how to think about whether an experiment without control is not a proper experiment and you lose your time. So these were my scientific mentor. Daniel was awesome in many ways, but he can look at science from outside and see the big picture. And I had the tendency at that time to get stuck with details. And sometimes you need somebody like that. Mentor I have right now here, Jacques Côté is also fascinated by all the fundamental biology and just making it happen and do beautiful science out of it and keep us track. And at the end, when you focus on fundamental question, that brings you often to mechanisms that are important, help you solve solution in either cancer or diseases. So that's something I admired him too, because he stick to this level. And then many women, because these are all men, right, that I've been having as mentor. During my PhD degree, I also had Jean-François Côté, who's now the director of the RCM, who was there all the time as a starting investigator at that time. So that was really a mentor for me in taking decision, really helped me orient my career in terms of postdoctoral fellow. I'm talking a lot generally, so I used to talk to a lot of people, have opinion and being able to do so that these are all day-to-day mentor. And for the woman, it's really the group that we have. I would say I could name people, but it's the group itself. I think that really helps and make me believe that you can be a good scientist and a mom in science at the same time. So these are the person I'm following. Locally, Sabine Louis is a really good mentor for me. She's passionate and she had her lab happen, even having kids at the beginning. So I guess I'm trying to find models and mentor that suit the life I'm doing, but still successful with science. So and there's plenty of it.
And having all those mentors, would you say networking is the biggest aspect to have that many contacts and collaboration? Yeah.
It's essential. I don't believe in one person science. I actually, when I joined science, I was a concern. Would I like to do science? Because it's you have the feeling that it's something you do by yourself. But there's nothing more exciting than having somebody else working on the same project and being excited of finding new ways to answer your question. This is true at the level of the lab, because when you have the student in the lab, you have these discussions and they become deeper and deeper as the student evolves in this environment. But also, if I need anything, any special tools, experiment, or expertise, I would grab, I would have to say the phone, but I would say I'll open a Zoom and chat with my colleague and really learn much more. And I think we move much faster. So in terms of collaboration, I'm really a highly collaborative person. In terms of networking, I think it's essential. It's the key of knowing what's happening, being part of a group. Also, it's important if there's a conference or opportunity that occur. This is really important. And to know who you're working with. There's many good stories and good people out there, and you would know them only by doing networking, because often a name on a paper, on a study, doesn't mean much, right? It's good to have a person behind it.
I was wondering if you had anything to say to graduate students that are just starting and don't have that many contacts or connections. What would you tell them? Like, if someone is shy to go, just talk to someone. Do you have any tips for them?
Yeah, I would say, and as I say, so when I'm training students, I always tell them that my job is to bring them in a position where they stay on the tip of their toes. This is not a comfortable position, but this is where you grow. So if there's, so here, where, for example, at our center, there's, when there's an invited speaker, you can go and have lunch with the invited speaker. This is a human being that like to talk, when they know you're a student. Go ahead, ask your question. You have no stupid question. It's like, what is it to do a PhD in the lab, or how does it work for them there? You don't have to talk about their specific, you don't have to have this brilliant question you think you need to have. So just go there, listen, and discuss, and you'll learn so many things, right? Maybe the way it's working in your lab is something you like, and you can see that it's not the same everywhere. Or maybe the opposite, maybe there's stuff you don't like, and you'll see how that works. I would say go ahead. In conferences, sometimes it can be harder. I would say try to go to smaller conference where PI are accessible, and you can also get along with all the students that are there. I mean, try to go and talk to the student. These are your colleague of tomorrow. So people you meet in the conference can, and I have many examples of that. When I postdoc interview, I met Alexandre Maréchal, who's now a director in the University of Sherbrooke, but we met through when I was postdocing, I mean, trying to find a postdoc. So this is just talking to people around me.
Have you found that, a lot of the people you were networking in the early stages of your career have also moved on now to similar stages as you now? So it's, it's not, it's like a natural networking progression that, then you have a lot of people who also have their own labs now, that you can collaborate.
Yeah. I think it's essential. I was talking with a finishing PhD student at the NIH a month ago, and she was a bit scared of continuing in that journey of having her own lab because she was a bit more social, and she thought it was an issue. And I was like, it's not, because this job is so diverse. You won't be the person at the bench anymore. You are at the beginning when you open the lab, but after, you're the person who, if a student have a problem, you're the person who need to find solution. You can do yourself, or you can use your contact, and you need to talk to people. You need to be out there to see what else is being done, right? And as I tell to students, you don't want to reinvent the wheel. If stuff has been done before, go and ask the person to do it. You'll learn much quicker. So I would say yes. A lot of the PI I know are a really social person.
Before just going on with your specific research field, why did you choose, obviously it's related to epigenetics, why did you choose this specific field?
I always been fascinated by how adding a small modification of the protein really regulates the protein. So when I was in PhD, I was studying how phosphorylation of the N-terminal of my favorite protein was regulating its shuttling to the nucleus and synchronizing its entry to the nucleus when the cells divide. And that's important for the virus to really synchronize its replication to the cell cycle. So that was the first time I was actually in contact with this type of modification. And I thought, so chromatin is really amazing. You can have this, you can decorate it with different modifications. So I know epigenetic include modification of the DNA and the chromatin that would actually allow the regulation of the genomic content without changing the genome. But I'm more interested in the part of chromatin. So how adding decoration of the histones really enable the recognition of this modification by factor that opens up the chromatin, close up, and really regulate the biological processes. How quick this is and how beautiful it is to have, because you would think of adding one modification, but there's actually a communication of all these modifications. How can it be simple while being complex?
And then I guess going into that in more detail, where the intersection of epigenetics and DNA damage occur, could you go into that in a little bit more detail? Like, how do these two, I guess, processes in the cell converge?
Yeah, so that's actually the subject of what we're doing in the lab. So my lab is divided in two kind of subject, right? There's part of my lab working on how viruses actually hijacking this pathway that are driving genomic stability in the cells and how they use it to their advantage. That's one thing. But the other is really try to understand. So there's many examples over the years. So first we know there's a break there's a damage, there's phosphorylation of a histone variant H2AX. So that's a hallmark of DNA damage. That's the first modification that is recognized by different factor that will be recruited to the break. And then when I did my postdoc, I was studying this E3 ligase that actually goes at the break and had ubiquitin up on double strand break. We know now this is also going into different types of breaks, such as the stress replication fork, for example. So E3 ligase also there. So there's also ubiquitination. And one of the big challenge right now in understanding the role of these modification in DNA damage pathway choice, this is the kind of the expression we use, is if you add a ubiquitin, and this modification on histone H2A, for example, is recognized by a series of factors that recognize the exact same modification. So how is that big? How does it allow the cells to decide? And there's beautiful study out there showing that, for example, you have H2A that is ubiquitinated, but you can have H4 that is methylated. And if the DNA is not replicated, it's dimethylated, and that will promote the recruitment of 53BP1. So it recognizes both ubiquitin and dimethylated K20H4. And that will actually promote non-homologous end joining. It's a type of repair that actually just ligate the two ends without looking for homology. So the cells kind of know at that moment the strands of DNA you have doesn't have homology. However, upon replication, the dimethylation is diluted into monoubiquitination, and that's allowed the recruitment of BARD1, which is a protein that promotes homologous recombinations. So the cell is able, with these modifications, to say this DNA is replicated, a template exists. I can use homologous recombination to repair faithfully the break. So this is where we are at the moment, understanding how the state of the chromatin is actually shaping the answer.
And you've mentioned homologous recombination. Are you interested in other pathways of DNA damage repair?
Mm-hmm. So we actually currently working on replication stress. So replication stress occurs when your fork slows down. And there's different things that can happen at that moment. If the replication fork slows down, cells will do anything to keep it there and restart it, because collapsing the fork is another level of damage. So there's different called DNA damage tolerance pathway there that would actually help the cells maintain the fork and repair either the nick. So there's different pathway there. And if the fork collapse, then you can use homologous recombination, template switching, fork reversal. And then if you look in textbook, if you start to read, it looks really simple, but then the more you read paper, the more complicated and the more gray it is. So different factors play a role in different pathways. So we're really working into this area right now. I try to decipher what's ubiquitination of the chromatin of histone and histone variant at the replication fork. How does it deal the response of the cells to this stress? And is it the same in different loci of the cells?
And then do you think in terms of like different contexts, like different types of diseases, different histone modifications might play more prominent roles in the DNA damage that might be occurring? So
yes, there is example that oncohistone, for example, would impact the choice of DNA repair. So that's one thing. So we're currently working on the histones variant called macro histone H2A 1.2, and this particular histones variant is really reduced in melanoma, aggressive melanoma. So you lose the expression. So there's two ways this histones is working. The first one and the most characterized one is that it's this histone promote repression of gene expression. If you remove it, that drives the expression of kinases that drives the proliferation of the cells. So this histone variant is really important to be there in specific location to prevent this to happen. If you remove it or lose it, it's associated with cancer development. But we also find out that in this particular case, there are different choice of DNA repair at the fork. And we think we can now use it to actually specifically target these cells that are deficient in macro H2A, and then try it. If you understand which pathway is triggering this condition, you can use different type of drugs to induce damage that will be more problematic to these cells to repair because they don't have the proper machinery. So there's plenty of examples like that where histones actually playing a role. With the occurrence too of all the drugs that are controlling histone enzyme that regulate histone deposition or histone modification, the more you understand about how this is working, the better it is in terms of improving chemo treatment, for example. So a lot of chemotherapy are based on DNA damaging agent, and you have all these series of inhibitor that would allow you to regulate histone acetylation, histone methylation, so that could really be combined to have a better impact on your treatment.
You've mentioned, well, your work is also on viral infections.
Mm-hmm.
So can you explain a bit more the interplay between viral infections and DNA damage repair?
There's actually two ways of seeing it, and depending on the virus you're studying, it's one or the other. First, when the virus enters the cells, the first thing you want to do is shut down innate immunity. So innate immune response. Innate immune response is mostly based on the detection of free DNA that is outside DNA. Part of the protein implicated in that is ATM, MR11, so protein that are also driving DNA damage response. So their goal is to recognize DNA hands, so free DNA hands. So viruses such as herpes virus will actually directly bind. So what we showed at the beginning of this year is that the IE1 protein of HHV6B, which is a type of herpes virus, is actually binding to NBS1. So it's a protein that will recognize the break and will inhibit ATM, the activation of ATM. So from the virus point of view, it's actually preventing the activation of innate immune response. So once you activate ATM, then you activate the response, the downstream response. But from a genomic instability point of view, your cells is also unable to repair its own break. And then every cells that divide, there's about 10 to 30 break every cell cycle. That's what has been estimated. So it's a double-edged sword for the virus. So first, virus want to inhibit this pathway. In other type of virus, such as human papilloma virus, there's a high level of replication that is required. So at specific time of the viral, during viral reproduction. So the virus infect the basal layer of the epithelium. So the virus will replicate with the normal replication cycle of this host cell. However, once the cells is migrating in the upper layer of the epithelium, then the virus change for what we call a amplification stage. And during that moment, DNA repair protein are activated, and they are required. If you use inhibitor for protein such as RAD51 or ATM, you actually kill viral replication. So the current model in the field is that since the virus replicate to so high level, they actually need this protein to cope with the replication stress induced in the host cell. So in that manner, this is how the virus could actually hijack the host cell machinery. So the virus itself is not alive. The only thing it has is a helicase and a protein that brings the helicase to the origin. So E1 and E2. Based on that, you need to replicate your whole genome. So you need to recreate the polymerase, RPA, all the machinery for viral replication, and all the machinery for DNA damage response. So we're studying what's the connection, right? So we know that viral proteins such as E1 will actually trigger ATM activation because of replication stress if it's unregulated. So as a PhD student, I showed that this shuttling is actually required to really replicate slowly and go out of the cells and prevent the damage to occur. So that's one of the mechanisms we show. And as a lab, we studied oncoproteins, so E6 and E7 from the viruses. And E7 is actually shown to bind our favorite E3 ligase, RNF168, and to actually reduce his ability to ubiquitinate it at double-strand break, and that's changed the repair choice of the cells. And that's really interesting because when people of the Memorial Sloan Kettering in New York study the signature of our infection, what they found is that these cells do more what we call microhomology engineering. So it's a repair that is based a bit more on the small homology. And it actually showed that when the virus is in cell, it's promote homology-based repair because we think of the impact on the ubiquitin. So there's different way we're studying how the binding of the virus to the the protein that is important in DNA damage signaling will have an impact on genomic stability, and how does this participate to cancer development, but also from the viral point of view, why is this useful? So if we deplete our favorite E3 ligase in infected cells, the virus cannot amplify. So it does need the protein. So how does that work? So we work on the two front, basically, on the three front, right? In cancer, on the viral replication, and in the inhibition of innate immunity, depending on the virus.
Can you just expand on the projects you're currently working on in your lab?
So in chromatin modification, we really try to understand how the S03 variant microH2A is actually shaping the answer to replication stress. So we actually produce chromatin in vitro. So we produce nucleosome in vitro with either canonical H2A or variant, and then we can ubiquitinate them in vitro and really see which factor is actually recognizing this nucleosome. And by doing that, we identify a new factor, RAD18, that is actually the role of macro at the break, prevent its recruitment. And we show in cells that the recruitment of RAD18 is toxic for the cell. So if macro is there, you prevent this recruitment and you repair properly. And if macro is not there, then you recruit RAD18. And this is toxic for the cell because you make the improper choice of DNA repair, which is toxic in specific regions such as telomere, ribosomal DNA, or fragile site, which are highly repeated DNA. So on the chromatin level, we're also studying remodelers that would actually promote compaction of the chromatin. We've shown that it's having impact on the level of the answer to the DNA damage. So you can see that if you remove this, this is going like crazy. You have a high level of DNA damage signaling, and this is actually improper for DNA pathway choice, DNA repair pathway choice. And in this particular project, we also found out that the viral protein is interacting with this protein. So we're studying how, again, it's promoting replication. So how chromatin remodeler are important for viral replication, and how this has impacted genomic instability in cells. So this is one of the projects. We use a lot of bio ID, again, turbo ID, to identify a partner of our favorite E3 ligase, either at the breaks or at replication fork. So at double strand break or at fork stall. So we can map the protein that are around. So we have identified new partner of the C3 ligase. So now we try to understand how does it impact its recruitment, its stability, its activity to ubiquitinated DNA, and its role in DNA damage response. So that's in terms of chromatin, that's basically what we do. We also use this turbo ID, so the fact that you can have the biotin in cells to visualize the protein. Where is your favorite protein? Because the E3 ligase we're studying are really expressed at low level, so all of the antibody don't work well. So we develop assay to be able to track the protein at replication fork. So we can test different type of stress, what are the protein recruited to, important for its recruitment, and what are the DNA damage response that it's trigger. So on chromatin and epigenetic, that's really what we are focusing on at this time. With the macro H2A, we're moving on to what happened in melanomas, right, where there's low amount of macro H2A. How can we use this information to study if they're more sensitive to different type of damage? So that is really exciting for that. And then on the viral point of view, we really study the interaction of at the replication fork, where the virus go at the replication fork. How does it change its speeds, its ability to cope with damage, and how is that important for the virus? So we're really playing on both sides using different type of technologies. And it's nice that students working on different projects, but they can really talk to each other because a lot of the projects are evolving around a D3 ligase that modify chromatin or being hijacked by viruses.
And then maybe, looking into the future plans that you have, are there any, techniques that you're looking forward to trying in the lab that are more, up and coming now?
I always go where science brings us. So I like to use technology that allow you to do big questions. So we use CRISPR screen, we use base editing. CRISPR screen based on the knockout, but also base editing to find the function of protein. So I forgot to mention, but we discover one new factor that act in complex. So it's ferment-figanin 1 that hack actually on the loading on Raf51 at DNA double strand break. But basically, this is regulate the response through double strand break and replication stress. And this is an obligate heterodimer. So we really study the biochemistry of this heterodimer at the moment. How does it participate to cell replication and DNA damage, and how do they exist as separate protein or together? So this is where the mechanism. So I'm really always bringing our research to the level of the molecular mechanism. This is what really it's exciting for us. So we take whatever we need to ask our question. So I'm not tool driven, but I use tool to answer the question we have.
And then, maybe going back to, the projects that you have in your lab, and then the PhD students who are doing those, how would you describe, your mentoring style with, as you said before, when you start your PhD, sometimes you really don't know.
I love this question because there's different type of mentality, and I often been told that I was too concerned about the well-being of students, but I know it's good. So what I do and I've been elaborating this way of training student with a postdoc that came in the lab 5 or 6 years ago, which is based on developing your competence as a person. And there's literature showing that as a PhD student, you need to evolve about 10 competence, which is going from how to look into research, which is broad research or small research, how to do experimental work, your ethics, your communication, your survival skill, which is perseverance, motivation, all of the above. So you need to develop these competences. So what we do is we established a form that we fill every year. So the student fill it and I fill it of why do you place yourself on that level of competence? Do you think you can do it by yourself? Do you think you need help? And then we discuss what you can use to help. So I really engage the student into identifying the strengths and the things need to be worked on and how I can help to work on. So this is how we work. It helps on many levels. It really helps on the communication. I mean, there's a lot of students who are highly talks a lot. It's easy. There's students that are more reserved, and these type of forms really helps. We recently updated with the university, and I think it's going to be really great because there's also studies showing where a PhD should be at the beginning or in the middle or at the end in terms of that knowledge. So that gives you a scale, and now we provide this scale to students so they can see, am I just help with anxiety? Am I okay? What I need to improve? I need to improve communication, but what do I need to improve? So there's different layout of things, and we can have discussions. So we do that every year. So it helps to develop as a person. So if you want to be a scientist, these competences are required. But if you don't want to be a scientist after, these competences are also required for other jobs. So how do you sell yourself? How do you use them on different jobs? So this is how I mentor. And I have an open door policy so people can walk in. Or I would say an open phone policy, because if I'm at home with students and there's anything, we have a Slack channel. Importantly, every month or so, I mentioned to student, I will answer you at 11 at night. I will give you an answer at 11. I don't expect you to answer at 11 at night, but this needs to be told. And we try to have a non-toxic environment. Science is competitive, so it comes with a lot of mental issue. And especially because when you're a PhD student, you go to class, it gives you things to learn, you learn it, you put it on paper, they say you understand, you have a good grade, this is good. You go into a lab, you ask a question, 95% of the time you're going to fail. Even if you have the best environment possible, mental issue, you're going to have a mental breakdown at some point because it's really stressful because you want to succeed, but it doesn't work. So how do you learn to deal with that? How do you develop the tools and how? So we discussed a lot about this, try to reinforce your mental strength and the way to cope with this, because this is often not due to the environment, but to the nature of the job.
Do you think graduate students are well supported to go through these, I guess, mental breakdowns, or whenever experiments fails? Science is a lot about success, getting your experiments, getting results. Do you think graduate students are well supported?
So I guess it depends where you are, but this is what we're trying to do here. I think it's norm that going in science, it will happen because it's not, unless you want to put your drug in a tube and add the IC50 and always do the same and try to find the best drug. I mean, this is going to work all the time. This is going to be more redundant. But if you go into a field where you ask questions about things that aren't known, you're going to face that. It's the same if you start your own business. You're also going to face all of these stress events. So the question is more how you develop your tools for me than what else you need. It's like you need to develop these tools. If you want to do this, you need to find your strength. And I always say, people have something else. Have something. Have a family. Have a hobby. Have something else, because when it goes bad, you need those to stay up. Because when science goes well, it's really easier, right? But you're gonna have this down moment. Make sure you're well surrounded, and then decide if it's for you or not, you know. And then there's whole mentality where you actually need to succeed in that field because you choose that field. It's great if you like it. If it destroys you, then you can change. You're not there to stick there because you start something. And I think training people by competence really help people understanding their value. You know, yes, your experiment don't work, but look at everything else you develop at the same time. So try to but I think that's where I
separate yourself from, you know, your worth isn't what the experiment result is.
Yes, exactly. And then you know, it may not work now, but we'll find solution. Maybe you need this 24-hour break because you cannot think straight, but then you'll come back after. Or so yeah, I think we would try to work on that. And I try to I mean, some personality have harder time. And then this is where I usually try to have co-mentor, co-direction. Co-direction, not when it's scientific level, it's needed, that's one thing, but also on personal level. Because it's not all the personality that fits, so I try to have another person that fits also, and that can help the person to have somebody else to talk to. And that improved the network too, so.
Do you think also, you know, doing your training in a smaller lab versus a bigger lab as well has like an impact on your development as a scientist, depending on your personality?
Yes. I would say you know what you don't like, but you don't know what you like. So for any decision you need to take, or choosing a PhD lab, or choosing a postdoc lab, or choosing a position in university or industry or anything else, go and visit and ask questions. I was convinced I was going at the Dana-Farber, right? I was going for, you know, I offer from Boston University, but then I walked in Toronto's lab, in Daniel's lab, and I felt that the team was there. And for me, that was more important than anything else, because I knew I would have a lot of fun with these people. And this is what the decision I took. I could have killed my career. People was saying, don't stay in Canada. You know, it's not true. It's not true. So don't do what you like. Do the best that you can. That would be the only, I mean, this is something I tell students also. If you want to do a master degree, finish that master degree, it's okay, but do it well. Just don't do it because you want a diploma. Do it well and open the doors. Every door is going to be good after if you did your best. So smaller lab for PhD was good for me because I opened the lab. So I really learned everything about, you know, how will we do a Western when we just received the equipment and nobody else around is doing a Western, right? So we try to improve to find solution. And this is it helped a lot improving network because we had to go and ask questions to others. So you improve on that level too. And big lab for postdoc was amazing in terms of scientific discussion. You know, it's another level, and it's also everybody is so pushing that you push yourself, right? So it's another environment. However, my personality, I knew that being at that level with family, I would have stressed out. This is why I took the chance of coming in a bit smaller university, but doing good science, but to a notch under in terms of the speed, because I knew I would be, and then it works for me. I'm not stressed as a PI. I have a lot of fun, and I believe that we do well, but just at a different speed.
So before we let you go, do you have any words of wisdoms for trainees who want to stay in academia?
Yes, I think this is an amazing job. This is the freedom of thinking and the flexibility. I know the freedom of thinking, you know, I always remember a couple of years ago was in a conference where everybody in the room talked about the mechanistic of the replication fork, you know. And then in the moment you're like, we're 200 of people talking about a small thing at 240. I mean, it's amazing. I can find people who actually have the luxury of having this thinking, right? And doing this research. So I think flexibility, freedom, stick to your values. And Daniel Roche told me that once, is always aim for the best and never cut corner for speed. And I think that's something, you should stick for. And why not trying it? You may fail or not. There's no guarantee anywhere. So I would say go for it if you want to try it, but don't avoid this career because you think it's too stressful to write grant. It is not. It is stressful to write grant, but it is to be in industry. It is stressful to managing a team in a business. It is. So yes, it is stressful, but you're well surrounded. You get better. There's a community out there. So ask questions and discuss.
That's great advice. So I think with that, we'll close and say it was a pleasure to host you, Amélie. It was really interesting to hear about your career and research. So thank you very much.
Thanks for having me. And then if anybody want to contact me for a question or anything, I'm always available.
Thank you for listening to Season 3 of the This is Epigenetics podcast. If you like this podcast episode, you can check out our website for future episodes at thisisepigenetics.ca.